Hello students, this is a short video where I'm going to clarify what is the basic difference between recombinants and transformants. Now, before we understand what is transformant and recombinants, the first point that you will have to understand is this is a vector, a plasmid vector, right? And this will serve as a vehicle to transfer our foreign DNA. This is our foreign DNA which we are going to incorporate into our vector, and then we will transfer this vector into the host cell, right? Now, when we do this process, that is when we are trying to add our foreign DNA into the vector, there is a possibility that some of the plasmid will get or will incorporate the foreign DNA, and some of them may not incorporate. The reason being, see, we cannot see this process. We cannot see a plasmid, right? We will just add all those things into the test tube. We will add enzymes. We will add foreign DNA, and we assume that some of the plasmid will get our foreign DNA, and there is a possibility that some of them may not get it. So basically, those who attach the foreign DNA are called as recombinant plasmid, and those who do not incorporate our foreign DNA, the one which are remaining as such, they are known as non-recombinant plasmid. I hope this much point is clear. Simply, if foreign DNA is added into the plasmid, we call them as recombinant plasmid. Whether if it is not added, in such cases, we call it as non-recombinant plasmid. So these 2 points are very clear to you now. Let us shift to the next point now. Now, this is our host cell, right? This could be a bacteria or anything. So this is our host cell. Now, when we add the plasmid into this host cell, there are three possibilities. First possibility is that the host cell does not accept the plasmid, right? So this is our host cell, which is as such, it has not accepted any of the plasmid. So it is as such, the way it was before, it is similar to it. So we call it as non-transformant because there is no change that has happened, isn't it? The way it was before, it is the same way now because it did not accept the plasmid, and therefore we call it as a non-transformant. Here, no transformation has taken place. Transformation means change. So here, no change has taken place. So we call it as a non-transformant. Secondly, what in case if the host cell receives the vector? Now you know that there were two types of vector. One was recombinant and another one was non-recombinant. So there is possibility that if it is receiving the vector, either it will receive this one or it is going to receive this one. So in any of the case, whether it receives this plasmid or this plasmid, we will call it as a transformant because some change has taken place, isn't it? Some change has taken place. Firstly, it was not having plasmid. Now it is having a plasmid. So some change has taken place, and therefore we will call it as a transformant, right? Now, the question comes, which plasmid it has taken? So in case, if it has taken a recombinant plasmid, then we will call it as a recombinant. Okay. Then we will call it as a recombinant. And in case if it has taken a non-recombinant plasmid, then we will call it as a non-recombinant. So even though it is a transformant, but it is a non-recombinant because the plasmid that is taken over here, that does not contain a foreign gene. But why do we call it as a transformant? Because it has received the plasmid. So therefore, this will be a transformant but non-recombinant transformant. And this will be a transformant and recombinant. I hope this is very clear. Once again, I'm repeating, there are two types of plasmid, recombinant plasmid, non-recombinant plasmid. The plasmid with our foreign DNA is a recombinant plasmid. The plasmid without our foreign DNA, in which the attachment failed, that is known as non-recombinant plasmid. Now, when you insert the plasmid into the host, there are three possibilities. Either it does not receive the plasmid. In such case, we call it as a non-transformant. Or either it receives the plasmid. In such case, we call it as a transformant. Now, if it receives the plasmid, as we discussed, there are two types of plasmid, recombinant and non-recombinant. If it receives recombinant plasmid, then we call this as a recombinant cell. And if it does not receive the recombinant plasmid, we call it as a non-recombinant. So this one is a transformant, but which type of transformant? Non-recombinant transformant. So I hope the confusion between recombinant, non-recombinant, transformant, non-transformant, everything is clear. Thank you.